Biological oxidation is the fundamental process that powers all living cells. To understand this process, let's look inside a cell where this vital energy generation takes place. Inside cells, mitochondria are the primary sites where biological oxidation occurs. The process begins with nutrients like glucose, which cells break down to extract energy. Oxygen plays a crucial role as the final electron acceptor in this process. During biological oxidation, electrons are transferred from glucose to oxygen through a series of controlled steps. This electron transfer releases energy, which cells capture and use for various functions. The energy released powers essential cellular functions like growth, movement, protein synthesis, and active transport. Through biological oxidation, cells can capture about 40% of the available energy from glucose, making it a remarkably efficient process. The electron transport chain consists of protein complexes embedded in the inner mitochondrial membrane. Electrons enter the chain at complex 1, NADH dehydrogenase, with high energy. As electrons move through complex 1, their energy is used to pump protons across the membrane. The electrons then move to complex 3, cytochrome BC1, where more protons are pumped. Finally, at complex 4, cytochrome oxidase, the electrons reach their lowest energy state and are transferred to oxygen. This process creates an electrochemical gradient across the membrane as protons accumulate on one side. The energy stored in this gradient will later be used to produce ATP through ATP synthase. The proton gradient across the inner mitochondrial membrane is essential for ATP synthesis. Protons accumulate in the inner membrane space, creating a higher concentration compared to the matrix. ATP synthase spans the membrane, acting as a molecular motor powered by the flow of protons. As protons flow through ATP synthase down their concentration gradient, they cause the central stalk to rotate. This mechanical energy drives the synthesis of ATP from ADP and inorganic phosphate. This process demonstrates how the cell converts the energy stored in the proton gradient into the chemical energy of ATP. This process, known as chemiosmotic coupling, links electron transport to ATP synthesis through the proton gradient. This process continues as long as there is a proton gradient and ADP plus phosphate available. While oxygen is the most common electron acceptor, many organisms can use alternative molecules in anaerobic conditions. Bacteria that use nitrate as an electron acceptor perform denitrification, converting nitrate to nitrogen gas. Electrons flow through different transport proteins depending on the final acceptor. This affects the amount of energy that can be harvested. Each electron acceptor pathway yields different amounts of ATP. Oxygen provides the most energy, followed by nitrate and sulfate reduction. The energy yield decreases as we move from oxygen to alternative electron acceptors, but these pathways allow organisms to survive in oxygen-poor environments. Cells carefully regulate biological oxidation through complex feedback mechanisms. Several key factors influence the rate of oxidation and energy production. The efficiency of glucose metabolism can be measured by ATP production at each stage. Allosteric regulation plays a crucial role in controlling metabolic rates. Environmental factors significantly impact the efficiency of biological oxidation. Let's review the key points about regulation and efficiency of biological oxidation.